The overall goal of this procedure is to describe a yeast high-throughput 2-hybrid assay that reveals interactions between the Pregnane X Receptor, or PXR, and the small molecule inhibitor Ketoconazole. This is accomplished by first constructing the yeast 2-hybrid plasmids, one expressing the human PXR ligand binding domain, and the other expressing the steroid receptor coactivator 1, or SRC1. Presence of ketoconazole disrupts the interaction of human PXR with SRC1 as revealed by liquid beta-galactosidase assays together with colorimetric screening of colonies on a filter. Screening of PXR mutants in the presence of ketoconazole allows identification of mutations that render the receptor immune to the action of the drug. Ultimately, LAC-Z expression by visual inspection and results of the liquid beta-galactosidase assays are used to show changes in PXR activation in the presence of ketoconazole, which allows the identification of original residues involved in the interaction.
We first had the idea for this method in Dr. Sridhar lab, where I'm an associate. When we were confronted with incomplete structure data on PXR, the biochemical techniques would not allow us to perform residue identification, especially for sites of interaction that were very shallow and not readily amendable to structure characterization. Based on the principle of detecting ketoconazole-resistant PXR mutants, we came up with an assay that would be tractable, feasible, and could be performed